Okay, everybody, this is my review for grade 10 biology culminating task for my students. And I'm sorry I couldn't be there with you to do this review, so this is the least I could do for you. So the idea here is to watch this video, and you're welcome to flip through your notes at the same time, try to find some of the same images, although I'm going to show you some different images as well. But if you want to follow along, you're welcome to take a couple of notes if you want for some of the question styles. It's not necessary. The main thing to do is to pay attention to the review and to participate and try to answer some of the questions that I'm going to ask you, whether you're answering them silently in your head or you're sort of talking about them with your colleagues. For any teacher who might be using this, there might be opportunities for you to pause the video periodically and let the students talk or get their feedback, this kind of thing. All right, so I just want to remind you that your notes are not allowed for the lab or practical component when you're going to be going around the classroom. Of course, we're going to be writing this in a different room, not in our normal classroom, in another classroom, and I'll put a notice up on the door about that. I'll remind you about that. But you're going to be doing a lab component where you're going around the room looking at microscopes and models and dissection specimens and all of that kind of stuff. And you can't use your notes for that. So you're going to have to memorize that stuff, know that stuff. You should already know it anyhow. But you can use your notes, but not the textbook, for the written part, the seat work. All right, so there's two parts to this culminating task. And you might be starting with the lab part, or you might be doing the seat work and then switching. All right, so let's move along here and see what I got for you. Okay, so we got a picture of the cell here. Let me just fix this up. Pointer options. Okay, so we got a picture of the cell here. This is actually a different one than one that you may have seen before. And the question is going to be to name the parts of the cell. So we're going to start up here with structure A. All right, and as you can see, A is pointing at this region of the cell right there. So can anybody identify that? If you can, just yell it out. Right, I hear you. This is the cell membrane or the plasma membrane. And just remember that this structure is semi-permeable, right? It controls materials that are entering or exiting the cell. All right, so that's the cell membrane. If we move on to structure B, you may have a little bit of difficulty with this. I'm not sure. You're looking at these little dot-like structures here. But will you keep in mind that these dot-like structures are also present over here? So does that give you a hint? What are they? Yes, those are the ribosomes. All right, ribosomes are often found adhering to this structure, which we're going to identify very soon, or they may be floating freely. Do you remember where ribosomes are made? If so, yell it out. Do you guys all agree with each other that ribosomes are produced inside the nucleus? Now, this is not that region. There would be another region right in there. That would be the nucleolus, and the ribosomes would then go through here. What's that little hole called? There, right? Nuclear pore, right? So the ribosomes would exit that and either adhere to this structure over here or just floating freely. What's the function of ribosomes? They produce proteins. Good stuff. Moving on to part C. Rod-shaped organelle with these lines that are perpendicular to the length, all right? Okay, there's one there, there's one there, there's one there. Mitochondria. One would be a mitochondrion. More than one are mitochondria. They produce energy for the cell. They're used in cellular respiration. Okay, we are on to D now. This wavy-like structure with these dots. This stuff would, of course, move all throughout the cell. Short form, long name, and you have to know both of them, endoplasmic reticulum. So this is the rough, so sometimes you put an R there, rough endoplasmic reticulum, which is a transport tube-like system for materials to move throughout the cell. Now, don't make the mistake of saying that ribosomes move through the ER. They don't. Ribosomes stud the ER. Ribosomes make proteins which then go into the ER and travel through the ER, but the ribosomes themselves don't travel through the ER. Now, I might as well jump over to here. All right, let's look at H. What's the difference between D and H? the fact that H is lacking the ribosomes. So what are we going to call this? Right? Smooth endoplasmic reticulum. Same idea, just without the ribosomes. Very good. All right, over to E. What is your best guess for what that is? You notice these little X-like things inside here. Now, the thing that bugs me about this illustration is this black line. I don't know why the black boundary is there. That might confuse you and make you think that this is the nucleolus, which is not. These are the chromosomes. All right, so this whole structure here is the nucleus. These are the chromosomes in the nucleus, but they are not bound by any additional membrane other than the nuclear membrane or the nuclear envelope. So those are chromosomes which control the cell, right? They are the genetic material or the DNA, deoxyribonucleic acid. Okay, yell it out. What's F? Did you agree with each other? F is the cytoplasm, jelly-like fluid which suspends all of the other organelles. Very good. And G is either the nucleus itself or you might consider it to be the nuclear membrane. All right. Moving on. We've already done H, so let's come up to I. Notice the similarity between these two organelles, and probably the best way to tell the difference is the fact that these ones have the ribosomes. Of course, this one doesn't, and they do look very similar. All right, but because this one's attached to the nucleus and looks a little bit like a passageway, you know, we're sticking with ER for these. So this is going to represent the Golgi bodies, the Golgi apparatus. And you sort of get the sense of these flattened or stacks of pancakes and the fact that these ends are going to be pinched off to produce vesicles, which will transport materials. And usually those vesicles will float up and bind or blend into the cell membrane. I couldn't think of the word there. And then release the contents outside of the cell. Now, when we come up to J, it's debatable. All right. Usually we talked about two possibilities. So just yell them out. What are the two possibilities? That one and that one. This is going weirdly, isn't it? All right. This could be a lysosome. And if it was a lysosome, it would contain digestive enzymes. It could also be a vacuole, in which case it would contain water for the cell. All right. So we've done many of the parts of the cell. What parts are there that aren't labeled? Well, it depends if this is a vacuole or a lysosome. And do you remember what else is present in an animal cell, not present in a plant cell? That would be there. Used for reproduction. Centrioles, right? To produce spindle fibers. And of course, there could be others that I'm just missing right now. What about whether or not it's an animal or a plant cell and why? Well, we don't see the presence of chloroplasts, but it could just be that we just don't see them. We don't see any evidence of a cell wall, and we should see that, right? The cell membrane is more interior. The cell wall is exterior. We'll see that in a minute in another image. We don't see any centrioles, so that might make you think this is a plant cell. But it is an animal cell. It is roughly spherical or round-shaped and doesn't have the chloroplast or the cell wall, that kind of thing. Okay? Okay, very good. We're going to move on to the next image. Okay, name the parts of the cell. So let's just go through this relatively quickly, all right? Some of these are repeats. So if we look at structure A here, we can see it's pointing right in the middle at this region right there. So that's going to be the nucleolus. All right, so as I'm doing these, either yell them out or just think in your head whether you could identify them. The nucleolus will produce the ribosomes, which will travel out through nuclear pores, which may or may not be visible here. B looks like it's pointing at the line here. So we're either talking about the nuclear membrane or we're talking about the nucleus in general. And I'm okay with you calling it either. Now, C is pointing at the white space inside the nucleus, so maybe B would be the nuclear membrane, and C is going to be the nucleus. Or, I mean, there is a fluid which is called nucleoplasm, which is sort of analogous to cytoplasm, you know, out here, and collectively the two are called protoplasm. I don't know if we've ever talked about that, but the cytoplasm and the nucleoplasm are collectively called protoplasm. All right, that's not important that you know that right now, but I'm just telling you about it. So that could be considered nucleoplasm or could just be the nucleus in general. Okay, let's look at D. What do you think D is? Looks like ER to me, because we're looking at it over here and over here. And whether or not it's rough or smooth, we don't really care. Okay, now we need to distinguish between E and F, it looks like. All right, E is the structure that's a little bit more internal, and F is a little bit more external, and I hope there isn't another one pointing at the same thing. I don't think there is. So E is going to be the cell membrane, and F is going to be the cell wall. All right, the rigid structure that gives support and some shape and stuff like that to the plant cell. And notice that this is roughly rectangular. See, it's not perfectly rectangular. So, you know, this one is going to be a plant cell. G, cytoplasm. How about H? This is an important difference between plant and animal cells. We can see a large vacuole here, all right, containing water and dissolved ions and sugars and lots of other stuff. All right, so that's our vacuole there. Now, I, okay, well, let's distinguish between I and L. If you look carefully at L, I mean, they're both somewhat rod-shaped, but L has these little structures in them that kind of simulate those cross-hatched things in the mitochondria. And so I want to say that I is pointing at chloroplast, and there are many chloroplasts. They would be green in a real plant cell, and they perform photosynthesis. Down here we see another mitochondrion right there, all right? So we have chloroplasts for photosynthesis, a definite difference between plant and animal cell. If we come up to J, we are looking at an X-shaped structure. These are all going to be chromosomes, and this diagram is just trying to illustrate the fact that we have a replicated chromosome here with a centromere in the middle and the chromatids joined to it off to the side. All right, we'll talk about mitosis in a little bit. Let me clear this up. And then come over to, where am I now? K. This is a pretty arbitrary, this is a terrible label. It looks kind of like it's pointing at cytoplasm again, or it might be trying to point at these little dots. All right, and if it was pointing at the little dots, then we could assume they might be ribosomes. It could be a lot of other different things as well in there. There's certainly not going to be lysosomes. They're too small for that. All right, and we've already said that L is a mitochondrion, okay? In terms of things that are not labeled, there are lysosomes in plant cells. There is a region that produces spindle fibers. You might remember it's called a centrosome, not centriole. And there are other things as well that we never even talked about in this course. And it is a plant cell. Okay, very good. Let's move on. Okay, now, this is cell division, looking at mitosis, interphase, cytokinesis, all that kind of stuff. So the first one is to name the phases of mitosis for each of these things here. So we have the cartoon drawing here. We can see some chromosomes here. These are chromatids joined together by the centromere right there. You can notice that they're all lined up. So this, of course, is going to be metaphase. This structure here represents the centriole. That's a spindle fiber. All right, chromosomes lined up in the middle. How about this phase? What do you think that is? Prophase. All right, we see the nuclear membrane beginning to break down here. We see the chromosomes have already replicated. They replicated before that in interphase. All right, so interphase comes before mitosis. These are the 4 stages of mitosis. So we can see the replicated chromosomes, but they're not as thick as we see them over here. And we can see centrioles. There's one there, or a pair there, and a pair there. And it looks like they're sort of migrating to opposite poles, so they end up right about there. Okay, so that was prophase. Move on to this one. We can see the chromosomes separating. What did we call that thing at that point? That's a daughter chromosome. This is the other identical daughter chromosome. This one's being pulled that way by the spindle fibers, all right? And, of course, in our cells, we would actually have 46 chromosomes going to each pole. Now, I'm actually realizing that I jumped ahead. I should have looked a little further. This is really a better cell for anaphase, seeing the chromosomes early on in their migration to the poles. Because we see this, the furrowing of the membrane here, what did we call that process? This is cytokinesis, right? But cytokinesis occurs after, or roughly after, the stage telophase, all right? And of course, during telophase, a new nuclear membrane would form here and would form here. So really, you know, this one is certainly an anaphase. We could start to argue about whether this is late anaphase or early telophase, because I admit that you see a little bit of this furrowing, but you don't see the nuclear membranes. And remember, this is just the artist's drawing of these things. But if you came across that and you had these two, I really would suggest you go for anaphase and telophase, especially if you're going to have just 4 drawings and you got to identify the stages. You're going to go prophase, metaphase, anaphase, and the telophase. So that takes care of both of those. This is going to be an animal cell because we see the presence of the centrioles there. I can't think of any other, aside from the fact that it's somewhat circular, but we're really looking at no, we're not just looking at the nucleus, are we? We really are looking at the whole cell, and that's the nucleus. So because the whole cell is roughly spherical, we probably are going to go with the animal cell. Time out. Okie doke. Let's move on. Clear that. Oh, come on, clear that. Why are you not clearing? Time out there. Clear. There we go. For some reason I got stuck. Doesn't like coffee. Okay, let's switch over to a different color here. Okay, this is the real thing now. All right, so let's say in the culminating task, you're looking through the microscope or microviewer and you have to identify it. Name the phase of the cell cycle, and it doesn't have to be just mitosis. So if we come over here to A, all right, these are all the worm-like chromosomes. They've been stained with something looking under a high-power microscope, and you can see they are all roughly in the middle. So this is going to be metaphase. I hope you can see that green on the background here. Now we can see the chromosomes are being pulled apart. All right, so we have a cluster of chromosomes there, a cluster of chromosomes there. So this one is going to be anaphase. As we come over to here, I'm really pointing at this little line here. You can see a little line in the middle there. That's called the cell plate. This is a plant cell. Look at the roughly rectangular cells, all right? And you can see a nice cluster, possibly a nuclear membrane reforming there. So that's going to be telophase. And this looks like another anaphase over here, doesn't it? Yeah, that looks like another anaphase, similar to this one. And this one, you can see the intact nuclear membrane. You can't exactly see it, but I mean, it's such a nice circular structure. They got to be held in there. And because it's very grainy looking and not worm-like here, all right, because of that, then we can assume that the chromosomes are actually very long and thin and stuff. Remember the name for that? Chromatin. So this is going to be a prophase. Either a prophase actually or an interphase, sorry. In fact, if push came to shove, I'm going to change my mind. I'm going to go with interphase here, and I'm going to go with prophase. I can't really see that one over here because now I can see the worm-like chromosomes, but they're still in the middle, that kind of thing. All right, and it looks like in this case, we had a cell plate put down and they went back into interphase. And by the way, I can sort of see a nucleolus there. Okay, and there may be more than one. Okay, well, hope that's helping you with reviewing your mitosis. Just quickly, I wanted to show you this one. I'm going back to red because this is an animal cell. All right, circular cells here. And you can see that in A, the chromosomes, and they're very small, but you can see them, are roughly along the middle. And so this is going to be metaphase. And here you can see the chromosomes, a big bunch of them, but being pulled. This is going to be an anaphase cell. And I mean, something like this might be metaphase. Again, I know you're looking at that thinking it might be prophase, and I wouldn't argue with you over that. But this one here, this is going to be an interphase. You can't even see the chromosomes. All these ones are an interphase. It's not that they don't have genetic material. All right. It's just that you can't see it. I mean, these are small cells. All right. And the chromosomes are so fine, you can't see them. All right. So that's mitosis in an animal cell. Okay. Now, another thing that we did in the course, in the unit, was looked at various types of tissue. All right. And here's a sample of some of the ones that you've actually looked under the microscope and that you would need to be able to recognize. You're not going to have to recognize things like adipose, ligament, tendon. We didn't look at those, right? But all the ones that we did look at, I know you can do. So we're looking in this kind of region for this type of cell. So we want the major and the specific type. So what is the major type? Epithelial, right? Epithelial. What is the specific type? Squamous, all right? Notice all these squished cells up here, flattened cells. Okay, so there we go. Let's move on to B. What's the major type of tissue here? Connective and supporting. Okay, and what is the specific type? Well, how could you miss it? The tree trunks, right? This is bone. Over to C. What is the major type of tissue? Muscle. What is the specific type of tissue? Well, you only looked at one. And the hint, of course, is all the zebra-like lines that are perpendicular to the long axis. Those are the striations. So this is striated or skeletal muscle. It's voluntary. You can control it. All right. How about the major type of tissue here? It's a little tricky. Connective and supporting. What specific type? Notice it looks a little bit like a sponge. It's compressible and flexible. So this is cartilage. Found in the ear, nose, stuff like that. All right, down to E. Notice the long cells here with the cilia. I'm not tracing them very well at all, but you can still see that. So major type, epithelial. Specific type, columnar. So people say this is columnar epithelial. And how about this one? What's the major type? Epithelial once again, because we've seen all these. And notice the cells that form this nice interlocking pattern to create ducts or blood vessels or whatever. So this is cuboidal epithelial tissue. And finally, this one, G, this is nervous tissue. And this is the neuron with all its branches for communication. All right. The epithelial cells work well for covering protection type things, insulation to prevent too much water loss. Connective and supporting is for, connecting and supporting for the body. Flexible here, strong like a tree trunk. Muscle cells for contracting. They're long, cylindrical so that they can contract. And these ones are highly branched, of course, so that for communication. Okay, little review on tissues. You guys are good at that. Here's a little bit more on tissues. What's the major type of connective and supporting tissue there? Well, I sort of gave it away, didn't I? Connective and supporting tissue, all right? Now, this is blood, of course, all right? That's just a general name. So this is connective and supporting. And then when we ask for the specific ones, we want the specific types of blood cells. So you're looking at A here, this one here. That is a WBC, a white blood cell. Usually larger and nucleated, right? We can see nucleus, all right? Very strange looking nucleus in this one, all right? These ones, of course, are red blood cells. They're much more plentiful. You can see the white space in the middle because they dip in like a little donut, all right? Maximum surface area by losing the nucleus to carry oxygen, all right? We've done those. Which type of tissue is not labeled here? Well, you can actually see little pieces here. There's one. There's one. There's one. What's that? That's the platelet, right? Sticky so that it can clot blood. Okay, very good. Oh, and by the way, don't forget that all of this stuff, what's that? That filling liquid, that's plasma, right? So we got the plasma in here as well. Okay, cool. Onwards. Okay. We also did the systems. Now, of course, remember, this review, it's already been about 25 minutes, cannot cover everything that we've done over a month and a half or 2 months, all right, to get that into 50 minutes or an hour video. Okay, let's name the parts of the digestive system. Now, this is a different diagram. You haven't seen this one. It's going to be a little different. Oral cavity. All right, within the oral cavity, we've got physical and chemical digestion. Physical because of the teeth and the tongue crushing the food. And although this is not labeled, there's one there, there's there, and here. These are the salivary glands that release salivary amylase, the enzyme, into the oral cavity. Remember that amylase breaks down starch into maltose. So starch is a sugar. It's a polysaccharide, long chain of glucoses. Maltose is 2 glucoses. I can't remember if we went into the details of that, but that's the beginning of chemical digestion. What is number 2 pointing at? Well, just note that you've got a 2 there and you got a 4 there. This one's a little higher, so how about we go for pharynx for number 2, the throat region, all right? And then the food is going to come down here. This is the muscular esophagus, and it's going to contract and push the food down into number 3. This is going to be the stomach, all right? And look at how similar that is in shape to the frog stomach that you guys saw in the dissection, right? Sort of a J-shaped organ, or backwards J. Okay, physical and chemical digestion there, physical and chemical digestion in the oral cavity. All right, can you think of some of the enzymes in the stomach? What about this one? What does that one stand for? Pepsin, which digests proteins, breaks them down into amino acids. There's also this one. Do you remember what that stands for? Lipase, which breaks down fats into glycerol and fatty acids. There's also acid in the stomach, HCl, hydrochloric acid, which helps in digestion. There's mucus. There are other substances as well. So physical and chemical digestion going on there. Very little absorption of nutrients, even though we're starting to break it down to nutrients. The food is going to continue to come down here, and now we are into structure 9, into the small intestine. So it's going to go all the way around there. Lots of enzymes going on there. A lot of chemical digestion, not really physical digestion for the most part. All right. And, of course, absorption. And when we say absorption, we mean if this is the small intestine, out of the small intestine into the surrounding blood vessels. All right. That's what absorption is all about. Let me clear that up. So small intestine, and then, of course, as it travels through the small intestine, it will go into the large intestine. What's another name for the large intestine? Colon. And the material will travel all across here, around here, and down to there. All right. What's a major function of the colon or large intestine? Especially if I do that. Right? Reabsorption of water. You can say absorption or reabsorption of water back into the body, out of this, creating the feces. It also produces vitamins and does a couple other things, but we basically talked about this. Inflammation of the lining, colitis. All right. Inflammation of the lining can impair water reabsorption, meaning water stays in here, coming out, diarrhea. This is the appendix. We never really talked about that much. There's the rectum down there for storage of feces until you release it. And this is the anus. And then out we go. A couple of other things. Let's talk about this. Largest internal organ. What is that? That's the liver. Produces bile, which helps to digest fats. Stores it in structure number 7 underneath there. What is that? Gallbladder. Right? Stores the bile. The bile acts in the small intestine, not in the stomach. All right? Helps to digest fats. The liver also stores glucose, stuff like that. Detoxifies the blood. We also have another accessory organ right here. So the liver's an accessory organ. We also have this one underneath. This one is the pancreas. All right. It releases many digestive enzymes into the duodenum, right in there, into the small intestine, along with bile from the gallbladder. There are many enzymes that come from the pancreas. We didn't really talk about that. We talked about 2 hormones, though. Hormones are not enzymes. Hormones are messengers. They tell the body what to do. Enzymes do the work. Hormones say do the work. Okay? Like teachers, right? This one is the main one that we studied. Insulin causes blood sugar to be decreased. And it causes that blood sugar to go into the liver. So the liver stores sugar. Okay, so that's one of the hormones. I don't know if we did glucagon. If we did, then great. If we didn't, don't worry about it. Glucagon causes the liver to release glucose into the blood. So while insulin lowers blood sugar, glucagon increases it. Okay, so I think we've answered all that stuff. So let's move on. That's the digestive system. Do I have more on that? Yes, I do. All right. Well, you also dissected the earthworm, so let's look at the parts in the earthworm. Don't forget, this is a model of the earthworm, right? You've seen this before. The food's going to come in here into the oral cavity. There's a little lip on top of that. That happens to be called the prostomium. It's not a big deal, but you saw that. Anyways, the food is going to travel down through here, sort of underneath and through this structure. This is the pharynx. And then underneath here, all through here, we're going to get region A. Oh, nice circling job, right? A. Esophagus, right? So long muscular tube conveying the food down to the various other parts. We are going to get this first chamber here, right? And a second chamber, and then this one is going to go all the way down the worm. Okay, so that's how that's going to go. So how about that? Crop, gizzard to grind, intestine. So we got the pharynx up here. So we're going to have pharynx. Okay, and we're going to have that PECGI. All right, so there's a little comparison of the digestive system of the earthworm. Don't forget that as far as intestines, we're not calling it small or large. It's just the intestine itself. Here's another view just to show you a couple of other things in the earthworm. We've got the esophagus right here, and then moving into that's esophagus, moving into the crop right there. Don't forget that the earthworm has a dorsal blood vessel up here and a ventral blood vessel there. Blood is moving anteriorly in this one, and towards the front, in other words, and posteriorly towards the back in that one. And of course, the blood comes down through these pulsatile vessels. These would be considered the hearts. All right, and they're on both sides. They're on this side here and on the back side as well. So there's 1 there, 2 there, 3 there, 4 there, 5. So 5 pairs of these hearts. Now, that's not critical that you really know that for the culminating. I'm just talking about that. And by the way, this yellow thing represents the nerve cord. It's on the belly of the worm. And there it is as well here. It's a little fuzzy in this diagram, but that's the nerve cord. And the brain, of course, is up there. And some of you saw that brain. Very, very small structure. And you can also see on this one the ventral blood vessel like this, and the dorsal blood vessel, and the hearts. Okay, so there's a little bit of a review of the earthworm. Let's move on. Okay, respiratory system. Structure A. This guy's little square nose there. All right, so air, of course, is going to come in through here. That's the nasal cavity. It's going to be warmed and cleaned. There's ciliated epithelial cells there. And the air is going to come down. And what would you call this region? Sort of a common region between this tube and this tube. That's the pharynx. The food will go down this tube, that's the esophagus, but the one closer to the front of your body, okay, more ventral towards your belly side, is the trachea. So right up here would be the voice box, and then you'd come down, and this is the trachea, and it's moving down in the chest region, and there are rings of supporting tissue that gives it some flexibility, but some strength. So what are those rings there? Cartilage, right? So this is the trachea, that's structure D. We've done all these guys. All right, and then it's going to split off into the 2 branches. Now, the 2 branches aren't labeled here, all right? But do you remember the name for one of the branches? That's a bronchus. There's a left and a right bronchus. These are then going to go into the lungs. So region J is a lung, and F is a lung. Now, don't forget, although this is the right side of the diagram, this is the person's left lung. So the bronchus carries the air in, and then we get into all of these branches indicated by G. These are bronchioles, so the air is coming down. All these like little tree branches, bronchioles, and then culminating in these circular structures. I'll show you another one in a moment. These are really clusters. There wouldn't be a single one. There'd be a group of them like grapes. These are the alveoli. So that would be indicated by structure E, alveoli. Underneath the lungs is this muscle, which is the diaphragm. All right. And so that's getting into structure H. As the diaphragm pulls down, it pulls the lungs with it. That means the lungs get bigger, right? Something like that. When that happens, you have increased the volume in the lungs. So what's happening with the pressure? Is the pressure gone up or is the pressure gone down? Well, if you've got increased volume, pressure goes down. Less pressure means, it's not being pushed as much. So that's what allows air to come in. There's more room, more volume, less pressure. Air gets sucked into the system. When the diaphragm, though, springs back up and relaxes, when it pushes back up, that compresses the lungs, makes them smaller. So now you have decreased volume and more pressure in there. It's tighter, and that pushes air out. So this is inhalation and this is exhalation dealing with the diaphragm. But you also have ribs, which the lungs are connected to, the rib cage, all right? And you have muscles in between. And those muscles pull the rib cage up and out, and therefore the lungs get a little bit bigger again. So with the combined effect of the diaphragm and the rib cage, that's what increases volume and decreases pressure. And that you breathe in. When these chest muscles, these rib muscles relax, the rib cage falls and the lungs get compressed, decreased volume, increased pressure, breathing out. Okay, so in terms of the structures not labeled, we don't have the bronchi aren't labeled here. Okay, let's go on and have a look at another image. This is a close-up now of a bronchiole right there. Whoops, I didn't want that. There we go. There's a bronchiole right here coming down. All right, this is also a bronchiole. And then ending at that cluster of air sacs that I've got circled right there. All right, cluster of air sacs or a cluster of alveoli. Name the tiny blood vessels surrounding the alveoli. Do you remember the name for those? These are the smallest blood vessels of the body. So these are called capillaries. And in particular, these are called pulmonary capillaries because they're inside the lungs. All right. Now, why are some colored red and some colored blue? Well, blue represents blood that is deoxygenated or deoxy. And red represents blood that is oxygenated or oxy. And in this case, the blood is coming in deoxygenated, saying, I need oxygen quick. All right, goes around the alveoli, and as you know, oxygen diffuses from the alveoli into the bloodstream, goes onto the red blood cell, onto hemoglobin, and so oxygenated blood comes out. It's like, oh, good, I got my oxygen. Let's go back to the heart. This would go back to the heart to be pumped to the rest of the body. So deoxy blood in, pulmonary capillaries picking up oxygen, giving CO2, by the way. This blood would be low in oxygen, but high in CO2, so that you breathe out. The CO2 would go from the blood into here, and you'd breathe that out. And then oxy blood back to the heart. Speaking of the heart, you got to have heart. Okay. This is a different view than what you've seen before. Let me try to switch to black. Name the parts of the heart. Now, historically, students have trouble with this. All right, there's a lot of parts. All right, a lot of anatomy. Let's start way up here. A, blood is traveling this way. Let's also look at B, blood is traveling this way. Notice the colors, blue. All right. These are large veins. This one is above the heart. This one is below. Can you name this one? All right. And this one? That's an I. Superior vena cava. Inferior vena cava. All right. Very large veins carrying deoxygenated blood into, and you can sort of see a hole there, into this chamber. C. What is this chamber? This is on the left side of the diagram, but this is on the right side of the patient. Right atrium. Deoxy blood from the head region and the lower body into the right atrium. The right atrium squeezes and the blood goes down into this chamber, through this structure in D. This is a valve between this chamber and this chamber. AV, atrioventricular valve. All right. Now the blood is in the right ventricle. The right ventricle will contract, pushing the blood up in this direction. What prevents it from going back into the right atrium? The AV valve, which closes. Blood then travels through another valve, F. What's the name of that valve? There are 2 AV valves and 2 of these kind of valves, F and L. Semilunar valve. It's called a semilunar valve. Blood comes up into this blue blood vessel. Now, although this is an artery leading away from the heart, this is going towards the lungs, and it's blue. It's still deoxy blood on this right side. This will take it to the lungs, so this is a pulmonary artery, and it will split, and it'll go under here, pulmonary artery. Off to the lungs, pulmonary capillaries, and all that stuff, carry oxygenated blood back to the heart in here. H and H. Pulmonary vein. Pulmonary veins here, pulmonary veins here. Look at these little dots, these little circles indicating the opening of these blood vessels into this chamber at the top. Left atrium, oxy blood. Left atrium contracts down through this AV valve into the left ventricle. So there are 2 atria and 2 ventricles. Now remember, this is different than the frog heart, which has 2 atria and 1 ventricle. Looks like some happy guy there, right? Okay, never mind. Let me get rid of all that. Okay, so we're into the right ventricle. The blood will then go up through here, another semilunar valve. There are 2 AV valves and 2 semilunar valves under here into the largest artery. This is the aorta, and it's going to carry oxygenated blood up to the head like that and over into the arms. Okay? And then, of course, down here, okay, it comes all the way down. It goes down to the lower body, to your feet. There, your feet there. Yay. Okay? Obviously, some parts are red because they're oxygenated and other parts are blue because they are deoxygenated. Now, let's look at another view of the heart here. Okay? This is an outside view, an external view of the heart. And let's see what structures we can name one more time. Okay? So you see this nice blue blood vessel here carrying blood down into the heart. You can't see the chamber and up. So here we have the superior vena cava and the inferior vena cava. Okay, those are all going to collect under here. So this is going to be an atrium, or at least the covering of the atrium. All right, this is actually called an auricle, which is AU, auricle. This is the right auricle, because it's just a flap covering, you know, the atrium. It's like another little room, but that's fine. This is an atrium. We can call this the right atrium. Okay. Now, C is pointing at either the muscle of the heart, the myocardium, the cardiac muscle, or just the location of the right ventricle, which would be down there. And the septum would be somewhere in here. Okay. And then the blood would come up and go through this. So this is going to be the pulmonary artery, although it's blue. All right. It's going to go that way, and it's going to go this way over to the lungs. The blood is going to come back through pulmonary veins, although they're red, they're oxygenated, but they're veins, back into the heart, and they'd go into there. And that would be the left atrium. And then down into this region, this would be the left ventricle, somewhere in there. And then it would squeeze, and the blood would come under. And then there you go, into the aorta and to the rest of the body. Now, one of the interesting things that I wanted to show you here is H. And I'm pointing at this because this is a coronary blood vessel, because we talked a little bit about the fact that the heart itself needs a blood supply, and that these blood vessels can become blocked or occluded by plaque or cholesterol or something. And if the blood can't get through there, that causes the heart, that part of the heart downstream, to starve of oxygen or nutrients or whatever. And that can cause a heart attack. And we talked a little bit about an angiogram is a way of looking at blood vessels and looking for these blockages. We already mentioned that the frog heart differs in that it's a 3-chambered heart with 2 atria and 1 ventricle. And in terms of the worm, we also talked about that. Just the fact that it doesn't have like a single big heart like this with 4 chambers. It's got those little pulsatile vessels, along the length of the worm, those little round things there. There's 5 pairs of them. So definitely a different circulatory thing. But the blood of a frog and the blood of a worm also carries oxygen and glucose and hormones and ions and waste products, just like the blood of a mammal. All right, so you need to understand the similarities and the differences with respect to those. All right, let's see if I've got anything else. Okay, I do have something else I want to talk with you about. And this is not something that we've covered so far. This is the carbon dioxide experiment, and I apologize for not being there to do this with you. We'll have a look at this after the culminating, but you should be able to understand some of the basic concepts of this anyhow. Carbon dioxide, as you know, is CO2, and we exhale that in our breath, and that's probably the easiest way to get CO2. All right. There is a chemical called Bromothymol Blue, and the short form is BTB. It's a chemical indicator, and we'll be using a bunch of indicators in the chemistry strand. And this can detect carbon dioxide, because if you come down here, let's say this is a beaker, and it's a beaker of water. All right. If we add some drops of this chemical in here, it turns blue. All right, without any carbon dioxide, just add this indicator. It's a chemical. It's blue in color, by the way. You add it to water, and the water goes blue. All right, so that's there. If you then use a straw and you exhale right here, you're breathing out, and you exhale carbon dioxide as you breathe out, carbon dioxide is there, into the water with this BTB stuff, there are bubbles of CO2 going into the water from your breath. Over time, as you increase the amount of CO2, as you keep breathing out, like you're breathing out, just keep breathing, keep breathing, keep breathing. All right, not like breathing out and taking a breath and breathing out. Just keep breathing out. There's more and more CO2 every second or whatever, right? And it starts to turn green. And that's because carbon dioxide reacts with this chemical BTB to give a green color. It's called a chemical indicator. It detects the presence of carbon dioxide. If you were to continue to breathe out as much as you can, at some point there is enough CO2 there that BTB turns yellow. So it can go all the way from blue to green to yellow. So high CO2, it's yellow. Not quite as much CO2, and it's green. No CO2, 0 CO2, and it's blue. So this can detect the presence of CO2. So you can imagine that the more CO2 you put out, the faster this could turn yellow. You could time this, all right, and have somebody breathe out into a straw and time how long does it take to turn, well, do we have to care about the volume of water? Would it matter if I put 50 milliliters of water one time and I did the experiment, and then I used 100 mils and 40 mils? Or should we keep that constant throughout the experiment and then test how long it takes for CO2 to turn the bromothymol blue solution yellow? So if you breathe out through a straw and you start to time it, all right, you find it takes, I don't know, let's say, let me clear this off for a second. Let's say it takes 30 seconds, okay? It takes 30 seconds to turn 100 milliliters of water with 10 drops of BTB to turn it yellow. And then you do it again, and you do it again, and you keep this all constant, right? Because you want to control stuff. And the only variable is you breathing out into the straw at rest. What would happen if you exercised and then did this again? Same volume of water, same number of drops. Don't go changing the volume of water and exercising. You're changing two independent variables. If you keep all these controlled, the independent variable is the fact that you were at rest or you exercised. Now, if you exercised, would you expect it to take longer to turn yellow or less time to turn yellow? Well, let's think about this. When you exercise, you need a lot of energy, right? And you make a lot of waste products. And you know you breathe faster too, don't you? Like you're out of breath. So when you exercise, your cells are taking in glucose, using them for energy, and converting the glucose to CO2, which you breathe out. So if you exercise and then do this, wouldn't you expect this to turn yellow faster? Because you are essentially breathing out more CO2 in your breath, or maybe just breathing faster. All right? So with everything else constant, the independent variable changing to being at rest or doing exercise, you could get a different result, a different dependent variable, right? The dependent variable is what occurs, what you're looking for, what you're measuring. The change in the time, instead of 30, it took 20 seconds with exercise. All right? What else could you do? Let's see if I got something, I think, on the next page. I believe I do. Okay? Here's some other things. Exercise. Now, what about the number of straws? I don't know that that would do anything. You could try that. All right? If you use 2 straws instead of 1, would you actually get more CO2 in here, turning the Bromothymol Blue yellow? Or would you just be, splitting it up into, half of your breath is going there, half is there? I mean, it's something you could try, right? Okay. What about the volume of water? Notice here I drew this higher, right? If you used 200 mils of water, but the 10 drops of BTB, but everything else the same, not exercise, just at rest and all that, timing how long it turned to go from blue to green to that final yellow color, would it matter? Would there be a change? I mean, this is all an experiment, right? Your independent variable is the volume of water. Everything else is controlled. Okay? And how about this? Changing the temperature of the water. Okay, this is the water here. You've added your BTB drops to it. Would it matter if you change the temperature? I mean, one thing to think about anyhow is, when you boil water on a stove, the way you get all those water bubbles and they eventually evaporates, right? Well, carbon dioxide is a gas as well, and carbon dioxide bubbles in here. It's possible, and I don't know, you could test this, that if you heat this solution up and you exhale, maybe it would take longer for BTB to turn yellow. Why would I say that? Why would it take longer? Well, what if the CO2 was evaporating and not staying in the solution to react with the BTB? If you heat it and the CO2 evaporates, there's less CO2 to react with the BTB to turn it yellow. So maybe it would take longer. It's like, I need to get my CO2 in there, but it's bubbling out, so I better keep going, try to build it up in here before it evaporates. I mean, I don't know. This is just something that could be tested. All right? So the dependent variable then is how long it takes for BTB to turn yellow. I mean, that could be an independent variable. You might want to study something different. Controlled variables are all the things that you have to keep constant. All right? And I think you have a good sense of what that would be. All right? So I think that's possibly all I've got for you. That is all I've got for you here. So I'm going to shut that down. So I hope that helps you prepare for your culminating task. I, of course, can't go over everything. And I know this has been a long video, but thank you for your patience watching this. Let's do it. Let's just go. Show me what you know. And that's really all it comes down to. Okay, you can do this. This is your challenge. You have to do this. I wish you all the best of luck. Thank you for watching. See you again soon.